Our laboratory is involved in linear and nonlinear optics in cancer research. It is obvious reason cancers are the most common and serious diseases in world population and one of the main causes of morbidity and mortality, much higher than other diseases. Our laboratory consists of laboratory of Raman spectroscopy, laboratory of Raman imaging, laboratory of femtosecond spectroscopy, laboratory of infrared imaging. So we are able to study structural, metabolic, epigenetic, immunologic, and genetic research. And dynamics of biological systems in with femtosecond resolution. We also have the real-time in vivo neurosurgical Raman system in our laboratory to perform a Raman-guided in vivo brain optical biopsies. So our systems are able to provide high spatial resolution in SNOM microscopy far below the diffraction limit, strong signal enhancement, specificity of interactions, AFM topography, stiffness, adhesion, and high temporal resolution, femtosecond resolution. We study human normal and cancerous breast tissue, brain, neck, and head, intestine tissues. Also cell human lines, breast normal and cancerous epithelial cells, and brain glial cells, astrocytes, astrocytoma, glioblastoma, and medulloblastoma. In vivo animal models, brain response of drugs, temodal, erlotinib, trastuzumab, mRNA vaccines, amantadine, and photosynthetizer. Briefly, our innovations are related to Raman biomarkers, Raman optical biopsy, virtual Raman histology, histopathology, the real-time in vivo neurosurgical Raman methods. The reason is obvious. We want to visualize cancer in real time as Dr. Cancer. But today I am not going to talk about these innovations. I am going to concentrate on cytochrome C in cancers. Cytochrome C belongs to family of heme containing metalloproteins, and cytochrome C is synthesized from two inactive precursor molecules, apocytochrome C, a protein that is encoded by a nuclear gene and imported into mitochondria, and heme, which is synthesized in mitochondria, and cytochrome C is located in the intermembrane space of mitochondria and is released into bloodstream during pathological conditions. So cytochrome C is a key protein that is needed to maintain respiration and cell death, apoptosis. Here we can see the cytochrome activity in animal brain. These are our results. One can see characteristic bands corresponding to cytochrome C. And we know exactly what kind of vibrations are observed because cytochrome C isolated from other species were studied in many laboratories for many years. So life and death decisions are made by cytochrome C. Cytochrome C can be observed in spermatoids even before the life begins, life starts. This green area and this green spectra in the Raman spectra, characteristic for cytochrome, are clearly visible. The dual function of cytochrome C comes from the capability to act as a mitochondrial redox carrier that transfers electrons between the membrane-embedded complexes 3 and 4 and to serve as a cytoplasmic apoptosis triggering agent activating the caspase cascade. In the view of the results for animal brain, it would be extremely valuable to control cytochrome activity in humans. To help address the challenges, we studied Raman enhancement of cytochrome for ex vivo human brain tissues of highly aggressive cancers like medulloblastoma. Here we have the Raman spectra, excitation wavelength Raman spectra for human brain cancer for medulloblastoma. Note that exactly the same spectra are observed as for animal brain. The most significant resonance is observed for the reduced form cytochrome at 1584 with excitation at 532 nanometers. The results were partially published, recently published, and the excitation wavelength Raman spectra for human breast tissue were also obtained, and again the role of cytochrome is very high. This resonance Raman enhancement is due to SORET band and for the Q band. And we have to remember that the Raman bands of the reduced form, one in the lower part of the plot, are much higher than that of the oxidized forms, blue spectra. And again, the Raman spectra of the reduced and oxidized forms. To check whether the redox state of cytochrome C is related to the cancer aggressiveness, we use the Raman redox state biomarker represented by the Raman intensity of the 1584 centimeters to -1 and other Raman bands. And here we have the human brain and human breast, and one can see that the characteristic spectra of cytochrome increases with cancer aggressiveness. These letters G0, G1, G2, G4 corresponds to the level of aggressiveness. And here we can see the intensity of Raman bands, cytochrome Raman bands as a function of the grade, as a function of the aggressiveness of the cancer, and one can see that these intensities increases. And here is the ratio of the characteristic peaks of cytochrome, and again they increase with aggressiveness of the cancer. Here we have a brain tissue, and in the brain tissue we can see that the Raman intensity of cytochrome C increases with cancer aggressiveness, while for most aggressive cancer decreases, grade 4. We also performed the polarization measurements at different configurations without polarization analyzer and with parallel and perpendicular beams of the incident and Raman scattered beams. They provide a lot of information on symmetry bands and on structure and conformational changes. Circulating in blood cytochrome C level is suggested to be a novel in vivo marker of mitochondrial injury. Various existing techniques such as enzyme-linked assays, ELISA, Western blot, liquid chromatography, flow cytometry were used to estimate the cytochrome concentration. However, until now, no technology has proven effective for detecting cytochrome C concentration in specific cell organelles. Therefore, existing analytical techniques cannot detect the full extent of cytochrome C localization and inside and outside of specific organelles. In Raman imaging, we don't need to disrupt cells to open the cell and release the cellular structure to learn about the biochemical composition. To check if vibrations of cytochrome C can be used also as a pathology assessment in living cells, we analyzed Raman spectra of brain and breast cell cancer cells lines in vitro incubation. And here we can see in single spectra, in single cells, one can see the characteristic bands of cytochrome, and again, particularly for mitochondria and for lipid droplets. Here we have Raman intensity as a function of wave number for normal astrocytes, for glioblastoma, for astrocytoma, for medulloblastoma, and one can see that this characteristic ratio corresponding to the concentration of cytochrome C to lipids decreases with cancer aggressiveness. Okay, now let's look on the role of cytochrome C in human breast ducts with Raman imaging. A few words on structure of human normal duct. We can see that inside there is a lumen, this empty white area. Around we have epithelial cells, and around epithelial cells one can see extracellular matrix and fibroblasts and connective tissue, adipocytes. Our results demonstrate how cytochrome C is likely to function in cancer development. On the left one can see normal human duct, and on the right human cancerous duct. One can see the differences between them. We used cluster analysis and the basis analysis to find the biochemical composition, to find the localization of the characteristic components of the duct, exactly in the duct, in the extracellular matrix, in the lumen. And here we have very interesting comparison. On the left, we can see the normal tissue, normal duct. And on the right, one can see the cancerous duct. Let's look at the oleic acid. One can see a lot of oleic acid inside the epithelial cells, much less in cancerous ducts. In cancerous ducts, we can see no beta-carotene concentration. Let's look at the cytochrome C. Cytochrome C is observed at a small amount for normal cancer in the extracellular matrix. However, in cancerous duct, the lumen is filled with cytochrome C, and this is the reduced cytochrome. In the normal duct, this is the oxidized cytochrome. Palmitic acid, again observed in the epithelial cells of the duct, it is released into the lumen in the cancerous duct. Cardiolipin, very important lipid in cancers. It is localized inside the epithelial cells, in the normal cells, in the normal duct, and it is transferred, it is released into the lumen and into the extracellular matrix. So, in breast cancer duct, the cytochrome C, cardiolipin, palmitic acid are the main components inside the lumen of the cancerous duct in situ. The presented results show direct evidence that cytochrome C is released to the lumen from the epithelial cells in cancerous duct. In contrast, the lumen in normal duct is empty and free of cytochrome C. So we can summarize that in normal human ducts, we observe low concentration of cytochrome C in the oxidized form. Cytochrome C is unbound to cardiolipin. Effective apoptosis occurs, and effective respiration through oxidative phosphorylation occurs. In contrast, in cancerous human duct, high concentration of cytochrome C in the reduced form is observed. Cytochrome C is bound to cardiolipin and reduced apoptosis occurs. Reduced respiration through oxidative phosphorylation takes place in cancerous human duct. The 15 minutes I received to present this lecture is over, so if you want to see the full version, including exciting in pandemia results for cytochrome C versus immune system, please visit the website of Victor Abramczyk Memorial Foundation, and the address is given here. Thank you very much. I would like to now acknowledge the financing, the project by the National Science Center. Thank you very much for your attention. I would like to meet you all at the normal conferences in the normal times. Thank you once again. Thank you.